Hello, this is our video experiment for Practical 1. Spectrophotometry 1, Protein Analysis, by Subgroup 6. Here is the group members.
Hello, my name is Erika.
Hi, I'm Genevieve.
Hi, I'm Priscilla.
Hi, I'm Veronica.
Hi, my name is Jonathan.
The first material we use is micropipette. It is used to measure and transfer small volume of liquids. Next, we used distilled water. We used it as a blanker to dilute samples to calibrate the spectrophotometer. Next, we used the Bradford reagent. His chemical can detect the protein concentration by changing the color to blue. We also used BSA protein standard, which is to create a calibration curve for quantifying unknown protein. To make sure the chemicals are evenly mixed, we used vortex mixer to rapidly mixes the solution. Besides, a cuvette was used as a holder for liquid sample and placed in spectrophotometer. We used a spectrophotometer to measure the absorbance of light to determine protein concentration. Then, we used sterile pipette tips to prevent cross-contamination occur between sample. Finally, we used test tube to hold all the liquids needed for the experiment. The first part, a standard curve, was prepared by using BSA protein. Firstly, label all 7 test tube. By using a micropipette, pipette 4 ml of distilled water into each tube, followed by 1 ml of Bradford reagent. Then, BSA protein is added in microliter. 50 microliter is added into test tube 1 and 2, while 100 microliter is added into test tube 3 and 4. Then 200 microliter is added into test tube 5 and 6, but test tube 7 receives 0 microliter, serving as the blank. After adding the solution, mix thoroughly to ensure uniform mixture. After that, transfer 1 ml each of the mixture into a cuvette. Then, the absorbance of each tube is measured at 595 nanometer using the spectrophotometer with the blank serving as the calibration. Calculate the average and the standard deviation for each point and then plot a graph of the result with the concentration as the x-axis and absorbance of light as the y-axis. Not to mention, include the error bars, standard deviation, on the graph and add regression line. For part 2, the same procedure are repeated for the unknown protein sample. All 7 tubes are labeled. Each tube is filled with 4 ml of distilled water and 1 ml of Bradford reagent. Then, 50 microliter of sample A was added into test tube 1 and 2, while 100 microliter of sample B was added into test tube 3 and 4. Then 200 microliter of sample C was added into test tube 5 and 6, but test tube 7 receives 0 microliter of sample as the blank. Mix thoroughly to ensure uniform mixture. Transfer 1 ml each of the mixture into a cuvette. Absorbance of each tube is measured at 595 nanometer using the spectrophotometer with the blank serving as the calibration. Record the reading of absorbance for each protein concentration sample A, B, and C. Compare the reading to the standard curve plotted and calculate the concentration of the unknown protein samples using the regression equation. For part 1, the table shows average absorbance and standard deviation of absorbance for BSA protein. As you can see, 0.05 ml has an average absorbance of 0.007 with the standard deviation of 0. Next, 0.10 ml has an average absorbance of 0.082 with the standard deviation of 0.011. Lastly, 0.20 ml has an average absorbance of 0.111 with the standard deviation of 0.007. Hence, there is an increase when it comes to the average absorbance. As for the graph, we can see that as the volume of protein increases, so does the absorbance increase. This is because a more concentrated solution with more molecules can absorb more photons. In short, more molecules can lead to more light being absorbed. In part 2, the concentration of protein can be determined by using the linear equation of standard protein curve, y equals mx, where m equals 0.1471. From the equation, the following results can be obtained, with 0.05 ml has the protein concentration of 0.6254 micrograms per milliliter, 0.10 ml has the protein concentration of 0.4521 micrograms per milliliter, and 0.20 ml has the protein concentration of 0.7308 micrograms per milliliter. This is the graph for protein concentration against absorbance. From the graph, there is linear relationship with R2 equals 1. The linear also shows a straight line that increase linearly, indicating as the concentration of protein increases, the absorbance reading also increases. New learnings. First, we get to learn on the technique to pipette exactly the amount of sample in distilled water using a micropipette. Next, we also learned on to use the Bradford reagent and then plot a graph based on the concentration and absorbance of light. We also learned how to apply a regression equation when trying to identify the concentration of unknown protein and how protein concentration influences the absorbance. Challenges faces. The challenges that we faced are not getting an increasing reading in absorbance of light for 100 and 200 microliter readings in part 1. We have to repeat the experiment multiple times and switch the apparatus and also the BSA protein until our reading finally increases. We also faced the challenges to set up the graph in Excel with the readings having big gap, which makes harder to obtain a smooth standard curve. Suggestions for improvement. Lastly, some of suggestions is to practice pipetting before starting the experiment to get used to the technique and apparatus. Plus watch some tutorial videos on YouTube or TikTok on how to properly set up a perfect graph for the results.